the phenotype of the resistance, and then the biochemistry of insecticide resistance mechanisms, also the molecular mechanisms that are in our region. So I want to go to the actual mechanisms because our next presenters, Dr. Audrey and also Professor Lee, will tell us a lot about these subjects, very important subjects. But I will only limit myself to the situation of the insecticide resistance mechanisms in our region. Next, please. So I'll start with Iran. We have resistance to a whole range of different insecticides in our country, starting with DDT in 1957 to pyrethroids in recent years, in the last decade. And we have resistance to a whole range of all insecticides in those four main classes of insecticides. Next, please. Now, that slide was about Anopheles stephensi. Now, if we talk about other anophelines in our region, in Iran, we have resistance to different insecticides in all major anophelines in Iran. But as you can see here, the majority of the cases, the red area here, resistance, is again for Anopheles stephensi. Really, Anopheles stephensi is the front runner in terms of developing insecticide resistance, not only in Iran, but also in the region. Next, please. Now, this is the situation in Afghanistan, where I studied a couple of times with our friends and colleagues from National Malaria and Leishmaniasis Control Program in Afghanistan. And again, there, Anopheles stephensi is resistant to major insecticides being used from different classes, including organocolorine, organophosphorus, carbomates, and also pyrethroids. Only in Kunar province, we could see a 98% only in this chart, which is in red, mortality with bendiocard. Next, please. Now, this is the situation in the region with different countries. Of course, I have added some other countries like India and Sri Lanka, which are not from our region. But anyway, I would like to share the experience of these countries, especially India, where Anopheles stephensi is a major vector, and they have, I mean, our Indian counterparts, they have a lot of experience in biology, ecology, behavior, and also insecticide resistance and its management in India. So you can see from this chart that wherever we have the report, we have R, meaning resistance to major insecticides from those OC, OP, carbomates, and pyrethroids group. Next, please. So this is, I singled out Ethiopia and Sudan because Anopheles stephensi is rather new in these countries. So again, you can see here in Ethiopia, we have resistance to, you name it, these insecticides that I listed here with very low mortality rates. So for example, well, with DDT, seems normal, 32%, but with other insecticides like organophosphates and carbomates, they are, I mean, propoxyl bendiocard, carbomates, 21%, 23%, which shows Anopheles stephensi, even being new in Ethiopia, it is rather strongly resistant to insecticides. Next, please. In Sudan, the situation is not any better. This is a rather, I mean, not very new study, but the species is resistant. I'm sorry, this is Anopheles arabiensis. This is resistant to deltamethrin, to DDT, and also only susceptible marginally to bendiocarb. Next, please. Now, there are going to be 4 maps of insecticide resistance of Anopheles stephensi in the whole of its range. And you know its range is now from the Horn of Africa to Western China. So you know the range is really vast. Next, please. This is for organochlorine. Next would be for organophosphates. Next, please, would be for carbamates, and the next for pyrethroids. You can see even with pyrethroids, they have widespread resistance. Next, please. Now we have, Professor Lee and also Dr. Audrey would talk to us in details about the mechanisms of resistance. We have 2 sort of fold of insecticide resistance mechanism, metabolic and site insensitivity. Next, please. In the first category, we have enzymes, metabolizing enzymes. And in the second category, we have mutations, maybe mostly point mutations altering the site of action of insecticides, rendering them ineffective against insecticides. Now, with the methods we use for insecticide resistance mechanisms in terms of the biochemistry of it, we easily collect specimens, homogenize them using the centrifuge, and then mixing buffers and reagents and do the spectrophotometry, and then finally read the data and analyze the data. It's very easy. It's done in our lab. Next, please. Quickly go through the next few slides just to show the practicals that we usually do in our lab. Mixing everything, the homogenates and the reagents. Next, please. Yeah, quickly, next, please. Yeah, and then a couple of more slides to just to show how easy this done. Yeah. Yes, next, please. Now, with the molecular mechanisms, it's again, if you are doing the normal PCR, it's going to be really straightforward and easy. Again, we have specimens, we extracted the DNA from the specimens and prepare everything, mixture and reagents, and also do the PCR and gel electrophoresis, and finally reading and data analysis. Next, please. And this is the banding pattern we expect to see after PCR and gel electrophoresis. Next, please. Yes. Next, please. So when we detect the biochemistry and molecular biology of molecular mechanisms of insecticide resistance, we have to bear in mind the cross resistance they confer to each other. I mean, when we have esterases involved in resistance, they confer resistance to pyrethroids, to carbamates, and also mostly and more importantly to organophosphates. With monooxygenases involved, we have mostly resistance to pyrethroids, and also with GST, we mostly see insecticide resistance to organochlorine. And also with molecular mechanisms, we have KDR, which confers resistance to pyrethroids and DDT, and also altered acetylcholinesterase, which confers resistance to carbamates and OPs. Next, please. Now, this is the biochemistry of insecticide resistance in Anopheles stephensi from Afghanistan. I don't have time to go through all the details, only just to show you the alpha esterase, beta esterase, GST, cytochrome P450s, and altered acetylcholinesterase. You can see, for example, here cytochrome P450 in CONAR is 2.7-fold compared to the susceptible one, meaning that in this population, we have the mechanism of cytochrome P450 running. So then here is a place you can, in fact, use PBO-net, for example. So this is the importance of knowing the mechanism of insecticide resistance. Next, please. And also the molecular mechanism of insecticide resistance in Anopheles stephensi from Afghanistan. You can see here the KDR East and KDR West playing a role in insecticide resistance in some of the provinces in Kunar and Nangarhar. Please, next one. This is the a summary of insecticide resistance in Anopheles stephensi in our region, in Iran, in Pakistan, in India, and in Afghanistan. You can see the mechanisms here, both metabolic and also site insensitivity. For example, with Anopheles stephensi to pyrethroids in Iran, we found that we have MFO playing a role, P450s, and also general esterases, glutathione S-transferases, and the situation in other countries are as follows. I can't really go through all the details. The PowerPoint and the presentation would be in the OneDrive. So please, to all participants, to go through the details and then would know the situation in our region. Next, please. Again, this is two different classes of insecticides with different species of anophelines, from Anopheles stephensi, subpictus, and calicifacius in Pakistan to other species like arabiensis in Ethiopia and labranchiae in Tunisia. We have different sort of mechanisms playing roles in insecticide resistance, which are summarized in this table. So you can see really a whole host of different mechanisms involved in different in resistance to different classes of insecticide. Next, please. So this is again in Anopheles arabiensis in Sudan, where you can see P450s, esterases, and GSTs playing a major role. So you can see here 2 to 14-fold, or sometimes even more, with these classes of enzymes involved in insecticide resistance in Anopheles arabiensis in Sudan. Next, please. So I think I'm coming to the near the end of my presentation. That this is the molecular mechanisms of insecticide resistance in Sudan, where you can see the frequency of KDR only West they detected in Sudan to different insecticides, about 80% of the frequency of KDR allele in different regions in Sudan. So it's and it's really related to the insecticide resistance in this country. Next, please. So in conclusion, we can, I mean, based on my and Samira's presentation, we have malaria vectors in our region that developed resistance to almost all major classes of insecticides. Different mechanisms, including metabolic and site insensitivity mechanisms, are involved in the resistance. So then we need new molecules, new tools, new technologies, new formulations for combating the vectors of VBDs in our region. Next, please. Now, the recommendation, the first one is technical. We have to really, as Samira mentioned, and I would like to emphasize, routine monitoring of insecticide resistance, establishing sentinel sites to do the monitoring with high and enough adequate temporal and spatial coverage, undertaking intensity susceptibility test, and also synergist bioassays, and finally determination of the biochemistry and molecular biology of the mechanisms of insecticide resistance. Now I think I have one more and final slide, which is the recommendations for the managers. We need to have insecticide resistance monitoring and management plan and regularly update it. We need to build structural and capacity and capability for insecticide resistance monitoring and management in our country. And also we need to establish a regional institutional network to support training and execution of insecticide resistance monitoring, management, and also the mechanisms, and also enhancing the information sharing mechanisms. I think that's it from me. Next slide. Yes, some scenes of my beloved country to everyone. I think there are going to be a couple of more. Okay, thank you very much.